Thank you so much for having me. I think this is perfect. I'm hoping maybe I can tie in into the last two talks. So, kind of the microbial composition and what that has to do for human health. And then also kind of personalized lifestyle recommendations and how we can impact human health. And so, from Viome Life Sciences, and what we're trying to do is kind of digitize, decode human biology and prevent and reverse chronic diseases. Unfortunately, I'm probably going to take a little bit of a break from kind of the dementia focus. That's something we want to tackle at some point. We haven't quite gotten there, so I'll focus mainly on mental health, so depression and anxiety, and show you guys what we have so far, the platform that we've built, and then also know that we're always super open to collaboration. So keep that in mind. Oh, I'm sorry. Oh, there. Okay. So at Viome, what we believe is that the human body, right, is an ecosystem. So a lot of the past research, we've been just looking at these human components of human health and disease, when in actuality, it's so much more complicated, right? So there's a vast microbiome, and that includes the oral microbiome, skin microbiome, gut microbiome, and those microorganisms that are in your body, they actually contribute an insane amount to actual overall well-being. So in terms of the gene content that humans have, we encode about 23,000 genes, which means we can make about 23,000 proteins that do, a wide variety of different tasks. But the microorganisms in one person can encode for over 2 million different genes. And so the gene potential of the microbiome is absolutely enormous, meaning that there's a huge amount of different biochemical activities that those microorganisms are involved in. And thankfully now, microbiome research is really starting to gain traction. And we're seeing that a lot of those microbial interactions are actually super important for human health and disease. And so what we at Viome are doing is we're trying to create kind of more of a systems biology approach to understanding human health and disease, how humans and our microbiomes interact to actually either promote or counteract a specific disease. And so this is just kind of an example, very high level of kind of what we're trying to do. So you can see, for example, this bacteria, there's inulin. It takes inulin, goes through a bunch of different chemical pathways, produces butyrate, and that butyrate is then actually going to be used by human cells, and it changes gene expression, which has a whole bunch of different roles. It can decrease gut permeability, improve immune function, decrease inflammation, and overall butyrate has a whole bunch of positive health effects. And so it's these pathways and these interactions that we're specifically trying to focus on. And what we kind of tailor our science on is how we can impact these pathways and the outputs and inputs of these pathways through nutrition and lifestyle interventions. And so, for example, for this example here with inulin, artichokes are super high in inulin, right? So if we want to promote these pathways, we want to introduce foods that have these substrates for these microorganisms to convert into positive substrates, right, that are going to improve overall well-being. And this is going to be holistically, but also on a disease-specific level, right? So some diseases are going to show that this specific metabolite is harmful or beneficial, and we want to target that specific thing as well for that specific disorder. Unfortunately, it gets quite a bit more complicated because inulin can actually also be bad if you have the wrong microbiome, right? So if you have methanogenic archaea, for example, they're going to convert that into methane, and methane is going to have a whole bunch of deleterious effects, right? So you get increased inflammation, constipation, you get bowel discomfort. It's associated with obesity, IBS, and IBD, and a whole other cohort of different diseases. So it's about knowing these interactions, right? Do you have the right bacteria to create the right byproducts, or do you have the wrong microbiome? And then do we want to add or do we want to take away those specific substrates to kind of push you into health? And so what we're trying to develop is kind of what we call nutrition 2.0. So we're currently in nutrition 1.0, which are these kind of fad diets, right? We're just saying this is going to work for everybody. You should, do X, Y, and Z, and you're going to be healthy and you're going to be better. But what we found is that that's really not the case. A lot of things need to be highly personalized and are very unique to an individual's microbiome, their composition of that microbiome, what it's doing, and the interaction that it has with the host, with the actual human. And so we've developed Nutrition 2.0, which is data-driven. So we have very fancy computer people that I don't fully understand all the things that they do, but they've got lots of machine learned models and mathematical formula that kind of create this architecture and network of biochemical interactions that we can then leverage and learn things from with all of the different metadata that we've collected. And so our goal is really to prevent and cure chronic diseases and cancers and to continually learn and build upon this platform that we have. And so it's important here, apologies for the grotesque image, but it's important to, when you're doing this mission that we have, right, to choose the right technology. So there's a lot of different technologies that you could use to kind of investigate the microbiome as well as the human. And what we do, which we think is unique and the most important part of our technology, is that we look at RNA. And so RNA is what makes proteins. It's what's actually expressed. And what is actually, it's a window into what is actually happening in the body or in the microbiome at that point in time. And a lot of past research has really focused on DNA. And DNA is, unfortunately, it's very static. It's essentially the same throughout your entire life. And so, you can see in this example here, this is somebody with IBD. This is their colon. You can see when their IBD goes through remissions and it goes through flares. So you can be in a healthy state one day, and then a couple weeks later you're suddenly having a flare and you're going to be in that sick state with inflammation, which is here on the right. And nothing in your DNA, right, has changed between this healthy and this sick state. It's the gene expression that has changed. So something has pushed you into abnormal gene expression, which is causing this disease onset. And so you can actually see that here. This heat map is this is gene expression quantification, and you can see all of these different genes are differentially expressed compared to a healthy individual. So when you're sick, you're changing your gene expression, but you're not changing your DNA. And so that's why we think it's really important to be looking at RNA so that you can actually quantify these changes and see it on that time scale that's actually relevant. Because a lot of these diseases don't really have DNA mutations that are causing the disorders. And even if there are risk factors, right? Risk factors for DNA or for genetic diseases we found are risk factors, right? Somebody could have that mutation and never develop the disease. Somebody else has the mutation and does develop the disease. And so the question is, why are you being pushed into this disease state? And why are some people not being pushed into that disease state? And so what we want to do is kind of modulate the human and microbial gene expression primarily through nutrition, but also with lifestyle interventions to move away from this sick state and move it back towards a healthy state and modulate all of these different abnormally expressed genes. And so we have quite a product portfolio that we've introduced over the years with this aim. And so we currently have tests that we sell. These are all direct to consumer. You can buy them on our website. And so we have a gut test, we have an oral test, and then we have a full body test, which includes a gut microbiome testing, oral microbiome testing, and then also we do a blood test just from a finger prick blood sample, and that actually assesses the human gene expression, so the person. And all of these tests provide nutritional recommendations. We also have personalized supplements, so those have food extracts, vitamins, minerals, things that we've macronutrients that we've identified that are gonna benefit your microbiome and overall health. And then I'm not going to go into these for this presentation, but we've also developed a oral and throat cancer screening test. And so with just looking at the oral microbiome, we have very good accuracy in being able to detect oral and throat cancer, which is very exciting, and we're hoping to expand that to a future diagnostic portfolios as well. And so what we do is we essentially run a direct-to-consumer business, and we sell our kits, we gather metadata from our customers, and then we constantly kind of iterate on that process, and we learn from the molecular data that we're collecting from our customers. And so we have over 300,000 samples from customers and research participants. We do extensive clinical research where we're specifically collecting samples from people in specific disease cohorts that we're interested in. We take all of that data from all of those participants. We do machine learning to generate our insights, which allow us to identify biomarkers. And those biomarkers, we then try to reintegrate back into our product recommendations to specifically target whatever it is that we're looking for. And so just one example here, we did a study on glycemic response. So, right, when somebody eats something heavy in sugars, they're going to see a spike in their blood sugar. And traditionally, it's thought that there's a glycemic index that each food is associated with, and it's thought to be universal, right? So everybody's going to react similarly to these different foods. But we now know that that's not the case. People are going to react very differently to different foods, and it's a highly personalized reaction. And so you can see here, these are two different people. This is the blood sugar response in response to eating banana or bread. And you can see this person here, the red line is when they ate the food. You can see that they have quite a rise in their blood sugar after eating banana, but not after eating bread. And then for this person, it's the complete opposite. They don't really have much of a rise when they eat banana, but they have quite a big rise after they eat bread. And so you want to kind of be able to control these big spikes in blood sugar. And what we did is we ran this study. We created a machine learned model that had about 80% accuracy to be able to properly categorize foods based on them generating a blood sugar spike in an individual. And what we did then is we implemented that into our nutritional recommendations. So you can see for this person, this is actually me. Couscous is generally thought of as a healthy food, but for me, it's going to actually cause a blood sugar spike. So in our recommendations, it's an avoid food. And it says here, your data indicates that you're likely to have an increased blood sugar response to couscous. And instead, it says try substituting quinoa. So we know now that quinoa is not going to cause this. So instead, I should probably eat quinoa to kind of normalize my blood sugar. And so we present all of these recommendations. We have these food recommendations where we put foods that are on the avoid. We also give a reason as to why those foods are avoids. And then we provide superfoods, which are going to be the opposite, right? Foods that are going to reduce inflammation and improve overall well-being. And then we also provide a description as to why we put those foods in those categories. And we also have personalized supplements, as I mentioned. So these are much more specific nutritional ingredients that are not just, eat a broccoli, which obviously is very complex in terms of its nutritional composition. And so this is much more specific and allows us to more precisely modulate the gut microbiome and those chemical interactions. And so now I'm going to talk a little bit about the gut-brain axis, which is very complicated, very interesting, very exciting. And I'm going to specifically just kind of focus on depression and anxiety, walk you through kind of our whole start to finish, well, we haven't quite finished, start to almost finish program on being able to generate essentially a platform that allows us to improve symptoms of depression and anxiety. And then I also have a little bonus data in there that's not related to either of those two things. So the first thing that we did is we got from all of the customers that we have, they complete a very extensive, sometimes maybe a little too extensive questionnaire that's got a whole bunch of different clinically validated surveys. And so one of those is PHQ-9, which is a depression survey. And so from all of the customer data that we got, we have about 2,000 individuals that are depressed, about 8,000 people that are not depressed. And we created a machine learned model. This is just with the stool data, so just looking at the gut microbiome. And we were essentially able to identify features that are associated with depression. And from that, we developed specific nutritional interventions that aimed to essentially take the molecular features that are associated with those microorganisms that are associated with depression and try to essentially starve them. And then the molecular pathways that are associated with health, we wanted to feed those and promote those. So we're trying to limit the amount of harmful chemical byproducts and maximize the amount of health-related chemical byproducts. And that's all through just modulating diet. And so what we did is we did a couple different studies in order to kind of show or convince ourselves that this was working. And so this was the first study that we did. And so this was just a crossover study. So there's no control group here. So each participant, we measured their depression, anxiety, and this bonus is IBS if you're interested. We measured it before they started Viome, and then we measured it after they started Viome. And so it's roughly, depends, it says here the time between time point 1 and time point 2, but it's roughly 200-ish days. And so time 1 is their score essentially before Viome, and time 2 is their score after Viome. And so you can see for anxiety, we significantly decreased their symptom severity for all severe, moderate, and mild anxiety. For depression, we also significantly decreased their depression levels. For severe and moderate depression, we didn't quite reach significance for the mild depression. And then just if you're interested, IBS, we were also able to improve the symptoms with IBS for severe, moderate, and mild IBS. And so this was super exciting. And we said, okay, I think we probably have something here. So we decided to go for a slightly more rigorous study. And we looked at then a controlled effectiveness study. And so here we used specifically individuals that are using our precision supplements. So those are the supplements that I presented earlier. And then we compared those to control participants. So those are Viome customers who are not taking the precision supplements and who also don't have reported not adhering to our recommendations. And what we found is also significant improvements in depression. So this is specifically for depression. And so you can see here, so these are the time point 0 for the precision supplements. So there's 9.1% of individuals have severe depression, 22% have moderate depression, and then 68% moderate to severe, and then 68 have moderate depression. And then this is the control group, which has a pretty similar distribution. And then time point 1, this is going to be after the intervention. And you can see that a huge amount of individuals from the precision supplement group now improve and are asymptomatic. And just a small portion of the individuals in the control group report being asymptomatic. So there's a much smaller shift in improvement in the control group than there is in the precision supplement group. And so this was super exciting as well. It kind of showed us or gave us confidence that our molecular features that we identified in the intervention that we're actually administering is having a positive impact on these individuals' mental health. And so then we decided to launch a randomized placebo-controlled trial. And that is what I'll show next. This trial is still ongoing. So this is preliminary results. I don't have any p-values to show here. These are just trends. Things are trending to significance. You can see as well the ends are going to be quite small. So we're not finished with this, but this is just kind of what we've seen so far. And so here we have for depression, specifically, these are just individuals that are over 50 and they're also overweight. We have baseline, and then we have 3 months later. We have 3 groups here. We've got the control group, and so this is going to be a placebo-controlled. We've got a VCS group, which we can actually ignore for the sake of this, but this is a different nutritional intervention that we did that was more generic. And then this red one is VPNP, which is the Viome Precision Nutrition Program, which is precision and personalized to each individual. And so you can see from baseline versus 3 months later, there's not a huge improvement between either the controls or the generic recommendations, but we see trending towards quite an improvement for the VPNP. And then this is the same thing here, but this is with anxiety, and we see kind of a similar trend there as well. And then as bonus data, which we also thought was very, very interesting, we had some secondary endpoints. So we were measuring HbA1c as well as fasting glucose. And you can see for VPNP baseline, HbA1c and fasting glucose both decreased much more so than it did in placebo. And so this is really exciting. We're hoping we're expanding this study. We're going to get more data. Hopefully we end up having some significant results here for both depression, anxiety, as well as blood sugar control. And that's pretty much all I have at the moment. We're looking to kind of improve this, expand this to a lot of different diseases. Dementia is, of course, a huge interest to us. It just becomes quite a bit more challenging to find participants. So we've kind of started with these low-hanging fruits and also kind of to validate that our system is working and makes sense. But so we're always looking for collaborators, and we're excited to expand what we're doing.
Fantastic, really interesting. I have many questions. I don't know. Hassan has a comment, and Manfred, who's coming from the field of nutrition, I have also a question, but I leave maybe the word to Manfred. You want to comment?
Sorry, panel. Make it short. I will lump up everything.
Yeah, okay. Because Ryan, I'm not sure. Are you going to stay for the panel discussion?
I can, yeah.
You can. Okay, fantastic. No, I was just wondering.
No,
I understand you do the RNA sequencing, and this is really your kind of core investigation. How do you connect this to those metabolites that might influence then, brain function through this gut microbiome axis?
Yeah, so all of the RNA that we sequence gets kind of associated with these specific microbial functions. So with our bioinformatics analyses, we say, okay, all of these sequences are associated with this biochemical activity that these microorganisms are doing. And then we don't really do anything. We let the machine learning algorithms essentially create these models, and then those models will rank the features that are most important with whatever disease it is that we're interested in. And we'll look at those, and we have a very smart nutritional science team that are much smarter than I am. And they will look at all of those molecular features, and they'll actually look at them, right? They'll look at all of them. They'll look at research, look at papers, and they'll say, okay, this molecular pathway is associated with this, or is a byproduct of this chemical reaction as a result of eating this food or this nutrient, right? And then the question is, do we want to introduce that or do we want to remove that? And that's kind of how it informs the nutritional recommendations. So it's looking at these pathways. Do we want to add more into that pathway, or do we want to stop that pathway and kind of starve it?
And you have really an experimental nutrition there, because you're seeing the stark effects, and so the more you corroborate, you go on with the collection of data, the better is the outcome, right?
Exactly. Yeah. So we went through these 3 different studies kind of on depression, for example. And at each of those, we kind of reassessed the model because we had more participants, refined it, and got it kind of try to work better and better and better.
Yeah. We have maybe Hassan, you want to take the conversation for later, or you want to ask a question?
No, it's okay. I just made a comment about the couscous, so it's just
about the couscous, yeah. And I think
a joke beyond the rest. Thank you for this message, and I've learned a lot. And thank you for.
Maybe Hassan should get into a performance nutrition program with Viome, right?
Yeah, maybe I should focus
Absolutely.
on your diet focus, and then will help me to perform even better for the next Ironman in 2024. Thank you.
That's the goal.
But Hassan,
sorry.
I read your I read your note, and I presume you love your mother, so she should be tested.
Yeah, that's right. Yeah.
As you said, you wouldn't tell her, yeah. So I think you should test your mother.
Yeah, that's right. That's a good point. Thank you, my friend.